Good morning, everybody. It's a great honor to be here and to be able to kick off this very impressive two-day meeting. I took the assignment of being the first speaker of what's going to be an incredible set of speakers quite seriously by feeling like maybe I should set a context. And so that's what I'm going to do. And I hope it'll be helpful. I'm going to give you sort of a nice overview of the past, present, future of human genomics, which leads us into this era of genomic medicine. And as I put my talk together, I deliberately decided with the examples that I'm going to give, I would tiptoe around other speakers so that to complement what I know there's going to be some incredible talks. You don't need to hear from me about those topics. And so I'm going to try to sort of skirt around and emphasize some things that I don't think others are going to emphasize. I did want to set a historic context for this. I really do believe that there are two scientific fields that launched last century that are changing medicine this century. And as a physician scientist, it's what drew me into this. Let's recall that the field of genetics isn't really that old. The word genetics was coined, and the first use of it in the scientific press was in 1907. And it set up the circumstance to really try to study the science of heredity. A lot happened last century, and at about the halftime of last century, of course, was the discovery of the double helical structure of DNA. And then what followed after the 1950s then led in the 1980s to the launching of a new field and the coining of a new word called genomics. And of course, the reason genomics came to pass was because after the discovery of DNA's double helical structure, we saw, oops, I blanked it, didn't mean to, we saw a series of truly seismic advances in molecular biology and DNA science, from elucidating the genetic code to learning how to read out DNA, to learning how to manipulate DNA and amplify DNA and do all sorts of things with DNA. And what started happening then in the 1980s was a bit of a drumbeat of discussion that would eventually lead up to the Human Genome Project. And that discussion started with a meeting where this was first discussed in 1984, to then some monumental and influential editorials and perspectives that were written that led, as I told you earlier, to the launching of this new field of genomics. And then we got serious. Then the field got serious through two very influential reports. I think some of it might have taken place in this building, actually, where committees came together under the auspices of very prestigious scientific organizations and developed basically a plan for pursuing something like that will eventually be called the Human Genome Project. To do that, it required money. In the case of the United States, that money came after a very influential hearing in 1989, which then solidified a plan for creating a new component of the U.S. National Institutes of Health that would lead the U.S.'s effort in the International Human Genome Project. So let me really stress, 1989 was a seismic year in the history of science, I actually think in the history of humankind. Let me remind you that in 1989, things really changed forever. And we will always remember 1989 first for the fact that Taylor Swift was born, okay? So we have to kind of contextualize this fully. But also, it was the year that the Human Genome Project was about to be born. Okay, and I was an incredibly fortunate individual, as I was in the right place, the right time. You heard I was postdoc with Maynard Olson at Washington University, and they got one of the very first Genome Center grants. I was able to participate in the Human Genome Project as a clinical pathology resident, as a postdoctoral fellow, and the rest is history. What an incredible project that went on from 1990 to 2003, accomplished all of its goals, and by almost any measure was one of the most impressive scientific feats in the history of the world. And so in 2003, the project came to completion with a formal announcement that the project was over, with a heck of a lot of partying and celebrating that we got it done. And it left us this text of 3 billion letters ordered, roughly 3 billion, representing the first sequence of a representation of the sequence of the human genome. So that was remarkable, and that was 22 years ago. And we all immediately appreciated that the Human Genome Project, while it was incredible, and we celebrated the finish line, that finish line then became a starting line. Because while we all wanted to get a sequence of the human genome, we appreciated, for all the different reasons, that all the scientific immigrants who came from other fields into genomics, because none of us, including me, was trained in genomics. The field didn't really exist until 3 years before the Genome Project. We all had reasons why we got on that original starting line, and we all had reasons why we wanted to see the Human Genome Project be completed. So the various funders of genomics around the world, and even in the United States, actually, many of them went in different directions. But those of us who were trained as physicians and physician scientists, and those of us affiliated with the National Institutes of Health in the United States, greatly appreciated that the next step for the next journey after the Genome Project was going to relate to medicine and the improvement of human health. Now, this was a bit of an obvious thing. In fact, the press in some ways beat us to it, because as soon as the Genome Project ended, you were able to see that both the popular press and the scientific press were immediately juxtaposing the two words, genomic and medicine, to do the obvious and start thinking about genomic medicine. And that was really exhilarating, but it was also a little terrifying. Because while we could spell genomic medicine 22 years ago, it was a very blurry concept. There was a lot that needed to be done before any of us as physicians could actually use genomic information in any systematic way to improving human health. Now, we could certainly begin to define it, and there was a vision. Let's not treat patients on average, but rather let's use unique information about individual patients to try to tailor their clinical care and do this for prevention and for diagnosis and for treatment. But while we could define it, it didn't mean we exactly were ready to implement that. And so we had a pivot. And 22 years ago, the field pivoted. I can certainly tell you that the National Human Genome Research Institute, which I had joined in 1994, and saw when the Genome Project ended, the entire enterprise pivoted with a pretty significant focus on implementing genomic medicine. But we also appreciated that the starting line was just the beginning, and the finish line was much vaguer than get a sequence of the human genome. It was changing the practice of medicine, something we knew would take far longer than 13 years, and inevitably would take a lot of steps, and just like the Genome Project, we didn't know what all those steps were. But we also had just come off of the Genome Project, where we also had an audacious goal, had no idea really how we were going to sequence the genome, but every time you accomplish one of your steps, you just pull out your pencil and you check that step off your list, and you just keep going. But we also appreciated that this second journey in genomics was not going to be a sprint. It was not going to be one country or one discipline or really one funder. And it was probably going to be a long haul, much more analogous to a marathon than a sprint. And so off we went to pursue this. Now, from the perspective of NHGRI and at NIH, we were tasked with the obvious responsibility of leading the US's effort in trying to see health benefits from the Human Genome Project. And so what the Institute did, and first it was led by Francis Collins, and then I took over the Institute in 2009, was to basically use a similar strategy that was used during the Genome Project of developing strategic visions that would basically guide the field over the successive years. And that's exactly what happened during the Human Genome Project. And so over the course of 3 strategic visions, starting in 2003, that Francis led, but I was a part of the putting together of that original strategic vision that came out the day the Human Genome Project ended, and then we renewed it after I became director in 2011, and then renewed it again in the middle of the pandemic in 2020. And I would just tell you that everything that follows have their fingerprints all over these 3 documents as we proceeded to go through this effort of trying to get through this journey from the Human Genome Project to the realization of genomic medicine. So what has followed in 22 years? So I'm going to condense 22 years of progress into about a minute and then take us into the clinical realm. But we knew that we had to deal with each of these things in succession. First, we knew that sequencing was too expensive. That first genome sequence produced by the Human Genome Project cost about $1 billion. So 2003, NHGRI said we needed to be $1,000. And as you all know, we've gotten there. We've now reduced the cost of sequencing a human genome by more than 1 million-fold, and we can check that off our list. In fact, it's well under $1,000 now, and that has changed everything. It's also allowed us to go in and now sequence millions and millions of genomes. And increasingly, we don't even know what the numbers are because so many have been generated, and some are available, and some are not, and some are behind industry, and some are shared in databases, and so on and so forth. And we've gone from knowing about maybe 10,000 places in the human genome that people vary to now hundreds of millions of places in the human genome where people vary, and we have rich data resources associated with the catalogs of these genomic variants. And by the way, for each of these successive accomplishments, there is a consortium, and actually Julie started to mention some of the names of consortium, either funded by NHGRI, funded by other funders, or in some cases, major contributions from hundreds, if not thousands, of scientists around the world who have joined in the culture of genomics to not only generate data, but to share that data quite widely to help us continue on this progression towards the realization of genomic medicine. Of course, just knowledge of genomic variation isn't enough. We actually need to understand how the human genome works. I would contend we've profoundly advanced our understanding of the human genome, but boy, we've got a long way to go. Certainly, we know a lot about our genes in terms of numbers. We don't know very much about what most of them do, and the complexity of non-coding DNA continues to be a very significant challenge, but one that I think through many international efforts, we are chipping away and getting better and better views of an understanding of the genome. I have to give it a medium check mark. I think we have a long way to go, obviously, in understanding the full complexity of those 3 billion letters. Oh, and yes, we have begun to apply all of these tools to now study human disease. And I would contend we've made significant advances on unraveling the genomic basis of human disease. There's been monumental advances in cancer. We're going to get to that later in this meeting. I would contend when it comes to rare genetic diseases, that's been a true triumph in many ways, where we have seen some of the earliest fruit. Sometimes I like to give numbers as a perspective. The day the Human Genome Project, by the way, there's thought to be about 10,000 rare genetic diseases caused by mutations in a single gene. The day the Human Genome Project began, October 1, 1990, there were 61 rare diseases we knew what the gene was. Cystic fibrosis, Huntington's disease, sickle cell anemia, cystic fibrosis, okay. 61. Today, it's over 6,000. That's pretty remarkable. Still 4,000 more to go, something like that, but going from 61 to 6,000. Of course, that's rare genetic diseases, not what fill our hospitals and clinics. Those are more common diseases that have a multiple and complex etiology related to genomic variants and social and physical environment influences. Those are really important to untangle, and we've all seen the onslaught of genome-wide association studies as knowledge of genomic variants has been intersected with major statistical studies to try to draw statistical correlations to important human medical traits, such as diseases like hypertension, diabetes, cardiovascular disease, and so forth. Tremendous progress, hundreds of thousands of associations. Some examples where we've wrestled it down to a single variant, finding a new gene relevant in a new biological pathway related to an important common disease. But we got a long way to go if we're going to understand common diseases. But nonetheless, I think it's worthy of a medium check mark, especially for rare genetic diseases, but appreciating that we still have a lot of work to do with common genetic diseases. All of this sets up this meeting. And part of this meeting is about genomic medicine. And we now have vivid examples of where genomic medicine has been emerging. I mean, when I took over NHGRI as the director in 2009, some people thought we were decades away from actually implementing genomic medicine. And when we published our strategic vision in 2011 that called for a very aggressive movement towards genomic medicine implementation research, I and the Institute actually got criticized a little by some, maybe some of the same voices that criticized the Human Genome Project, but just said it was too soon. And it just wasn't too soon. And while I will only give it a little check mark, because I think these earliest examples are just the tip of the iceberg, I think they're worthy of a meeting like this, which is why we're here, but importantly, why I believe I'm very comfortable saying that we have now gotten to the point that in some settings, we are able to sequence a patient's genome and compare that genome sequence to a reference genome and come up with a list of genomic variants and be able to use that information to now implement genomic medicine. I'll come back to this slide later in my talk, but needless to say, when you can have an opportunity to do this, in some cases, you are able to implement genomic medicine. And you may ask, okay, what is the list of examples that it's here and now where genomic medicine is being implemented? And so let me unpack what are typically current in 2025, my list of examples. First of all, is cancer genomics, and here I'm going to change hats, because I was the teacher, but I'm going to spend the rest of this meeting being the student, because I know some of you, and you are experts in this area, and I am not. And you've lined up some terrific speakers who are going to teach us about how cancer genomics is leading to changes in the practice of medicine with respect to how we're dealing with cancer. So I'm going to put that to set up to some other fantastic speakers. I'll say a few more things, though, about rare genetic disease diagnostics, because I never thought this would really happen in my professional lifetime. But we really are here. We really are in a situation where a child can come in, or maybe even an adult in some cases, and it just seems that something is going on genetically. And instead of doing a lengthy diagnostic odyssey of working that patient up, earlier and earlier we just get a genome sequence, and we take that information, and with that we're able to get a diagnosis. And we don't get a diagnosis every time, but there's lots of articles, lots of meta-analyses, lots of reviews, but something on the order of 30 to 50% of the time these days, you'll get a diagnosis, and it will only get better as we understand the human genome better and better. And it's being implemented worldwide as part of standard of care for working up a patient with a suspected rare genetic disease, and I would point you to a review article that talked about the current state of implementation that was from this year. And you're seeing more and more of this literature accumulating. I want to tell you about something that I'm pretty excited about. I don't think anybody else is going to talk about this, and it directly came out of an NHGRI program that we implemented very early on after I became the director, and it related to newborns who were acutely ill in a neonatal intensive care unit led by an investigator named Stephen Kingsmore, where it became very clear that there was an opportunity that if you could rapidly get a genome sequence of a newborn, especially a newborn that nobody knew what was wrong with this poor little child, that you could save their life. And what's been remarkable has been the uptake in the period of about 11 years, such that there have been many, many studies where this is done. And by rapid, I mean like in a day or two. And there's now even review articles that came out last year that because of what has been implemented in the United States at some level 4 NICUs and also in the UK, there's been a number of studies that really show significant ability to make a diagnosis upwards of 30 to 40% of the time. But importantly, you get a diagnosis. In some cases, you change management, you get those kids out of the NICU quicker, and you get them into specialty care where at least you know what is going on with that child in a way that we believe will drive down the healthcare costs for those difficult cases, but also will provide better care or at least better clarity to the families who are dealing with these acute situations. This has actually been in the news this week. It was in the news last week, and it's been bubbling on social media ever since, because the idea is you've got to be rapid here. Some of these are very fragile newborns. You've got to be quick, and so there's a little bit of gamesmanship going on here. And there was a new world record that came out of the Broad Institute. It's actually it's online at New England Journal, where you could read, it's right there, the headline, is they were able to get a whole genome sequence done and analyzed in less than 4 hours, which is pretty incredible, and made a diagnosis out of that. And so now the Broad Institute is very proud of their Guinness Book of World Records. This is also finding interesting utility in pediatric intensive care units, and based on one study, in adult intensive care units, where in this study that came out earlier this year, at University of Pennsylvania, they looked at a whole set of adults who found their way into an intensive care unit who did not have an explanation like trauma or an existing disorder like cancer or something like that, where they sequenced their genome, and they found that 24% of those individuals were harboring a rare genetic disease that accounted for their fragility and why they were in the intensive care unit. In some cases, it changed their management. We might start to see genome sequencing as part of standard of care when it comes to patients admitted to intensive care units across the lifespan, and I think that's pretty interesting. Going on with my list, I would point out, and one thing we really won't talk much about, but it turns out, if you didn't know what is the #1 genomic test in the world today, I keep blinking this, is non-invasive prenatal genetic testing, where small amounts of DNA are detected in maternal blood as a prenatal screen, basically obviating the need for amniocentesis, chorionic villus sampling. As a screen, the uptake has been tremendous, in part because companies formed around it and commercialized it, and in part because insurance companies, at least in the US and other places, are paying for it, and in some countries are just part of their standard of care, such that it is now estimated that 8 million pregnant individuals will get a non-invasive prenatal test done this year. So that's the #1 genomic test to my knowledge. There's also been a lot of excitement. I don't know if we're going to talk much about pharmacogenomics, but obviously there's a lot of genomic variants that all of us carry that influence which drugs are the best ones for us to use among the list of common drugs. And there's good successes. There's been a little bit of a disappointing uptake, at least here in the United States. We're hoping to improve that. There's a lot of big studies going on to try to implement pharmacogenomic testing more broadly. And it's not all about medicine. It's also about prevention. And we are starting to see more and more examples of genomic-based prevention. For rare diseases, this is absolutely the case. For rare disorders, both in the pediatric period, also adults, certainly, and maybe some of our cancer researchers will tell us about this when, in terms of cascade screening of families, once an initial identification of cancer is found in one family member. And that's in the case of single genes driving disorders like cancer or nutrition defects and so on and so forth. But of course, the other big rage that's going on is the idea of polygenic risk scores. And will we get to a point where even if we don't know the etiology of common diseases, we will be able to scan across genomes, find enough information about variants, and stratify people to be able to make risk predictions for common diseases? I think that's still in the research realm. I put money where my mouth was when I was at NHGRI. We had major studies being done to study whether or not polygenic risk scores really will be effective or not, but I still think it's a research endeavor. I don't think it's ready for a routine clinical practice quite yet. So that's my list, which I still think is the right list for now. There might be others that are going to get on that list. But more importantly than anything else, what I'm so happy about is over the last 23 years, we went from this vaguest idea of what genomic medicine is all about to bringing clarity to the situation. It's still just the tip of the iceberg, but we now can see exactly for these examples what genomic medicine is going to be all about. But I do like to watch the horizon, and I do like to be provocative to get us to think also about the future. So I sort of told you about the past and the present. Let me just peer in the future, because sometimes I get asked, what's the next example of genomic medicine that you think is going to hit prime time? And I have to admit that if I go back to 1990 and I heard some of the rhetoric when the Human Genome Project began, and to try to generate excitement, people would say, maybe one day every baby born will get their genome sequenced, and that genome sequence will go in their electronic medical record and they'll be followed for life. And that just seemed like a great science fiction idea. It wasn't out of the question, but it just seemed so far in the distant future that we just wanted to get the sequence done for the first time. But still, the seed was planted. I did find it audacious that Time magazine in 2014, now 11 years ago, made a prediction in one of their future issues where they talked about things happening in the future. And they made the prediction, incorrectly, that 11 years from 2014, everyone would get their DNA mapped, imprecise, they should have said sequenced, at birth. And they made that prediction. And to some extent, I even think in 2014, I was like, wow, I can't imagine that's going to happen by 2025. And they're right, it's not happening by 2025. Do I think it might happen by 2035? Maybe. Just maybe. And I put this on my watch list. I think we should be watching very carefully, and the journals are doing it too, and the literature is doing it too, and the funders are doing it too, that maybe newborn genome sequencing is not that far away. And you're starting to read about it, and you're starting to hear about it, and some countries are just grabbing it. And I would point out that I think the UK is really leading the pack. I don't know if they're the sole leader, but I've been watching them very carefully. They have a major study. I think they're taking 100,000 or 200,000 newborns and healthy newborns, sequencing their genomes and following them. And they're starting to make some pretty bold predictions about maybe that they will, basically start sequencing every baby in the UK, the genomes, within a short period of time, maybe the next 10 years. I would also point out that there are major review articles already being written about this because there's so many studies that are now ongoing, and there are so many studies that a new organization has been formed called the International Consortium of Newborn Sequencing. They are meeting tomorrow and Friday in London. I went to their meeting in New York last year, and every little dot that you see on that world map is an example where there is a major study going on where healthy newborns are getting their genome sequenced, and we are taking a look at what those the consequences are and how to learn from it. For example, the one in the UK, which is shown at the top, is called Genomics England Newborn Genome Sequencing Program, and the one on the bottom is an effort led by Wendy Chung called the Guardian Study. These are just two of those dots, but every one of those dots has a website and an affiliated program associated with it. Of course, this is not without some ethical issues, and I would strongly encourage, as this gets discussed, to follow the editorials and follow the discussion. I think there are many ethical issues to deal with, as there always has been in genomics. But the NIH is getting excited about this, even though I had nothing directly to do with this. I had a little bit to do with this. But this is also breaking news, something called the Beacon Study, which is out of the NIH Common Fund, something called the Innovation Fund. And they decided to basically fund a major program to look at this. And the winner of this is a consortium led by Robert Green from Boston. And I just saw this on social media last night, so I changed my slide. It is important that this program is really taking newborn sequencing of healthy newborns out for a test right, doing it in the context of the public health system of the states who basically drive screening of newborns for genetic disorders currently, and we believe in the long run will be the place that this is going to be driven, is at the state level. So I would keep your eye out for the Beacons program, because this money just went out to these investigators, and it was discussed heavily at last week's American Society of Human Genetics meeting. I also thought I would share with you, because I find this provocative, and we're really going to be thinking about what's it going to be like if newborns start to get their genomes sequenced. And so Robert Green, who's leading this effort, no relation to me, by the way, but he recently also put out a TED Talk, and it was a good TED Talk. I strongly recommend you watch it, but I want to share with you the last minute and a half of it, because it just makes you think a little. So here's Robert. If you've never heard Robert, he's also very charismatic. So here's Robert in this TED Talk, the very last minute.
If we really want to invent the future, we've got to do something different. If we really want to invent the future, we've got to realize that a child's DNA doesn't change over time, but the science is changing all the time. And so what that means is we should sequence your child's DNA, and we should revisit and reanalyze that DNA over and over again to truly create the dream of genome-informed medicine. Because each and every year there will be new insights and new treatments available. Well, this isn't offered anywhere in the world, but I'm happy to tell you that we are trying to build this. We are building an AI-enhanced digital health platform so that you, your grandchildren, your children, your pediatricians, your healthcare centers, your employers, your nations can do this at scale. It's going to take a certain amount of courage to change the way we think about disease, to embrace the knowledge of risk in order to preserve our health rather than waiting for us and our children to get sick and treating them there. But if we can do this, if we can embrace this, we can save millions of lives and usher in an entirely new era of genome-inspired medicine. Thank you.
So I am going to quickly, I'm not going to give the last set. I had challenges I was going to tell you about. I'm not going to do that. We can do that during the discussion, because I don't want to go over time. But there are challenges, and I'm happy to discuss them. But in closing, I just want to make one more point. Over 35 years, we've seen a magnifying glass brought to the human genome. And after the Human Genome Project, we had these threads of genomics get woven into this incredible tapestry that I described over the last 20, 25 minutes, increasingly first basic science and then human genetics and increasingly clinical applications of genomics. And we will continue to weave that tapestry, and that's really remarkable. And I've been just so delighted to be a part of that and so proud of what NHGRI contributes to that effort. I am still so amazingly proud to be a part of this incredible community of scientists that at first were very small when the Genome Project began, but now when I think about all of you were part of this, and so many thousands of others who are just standing on the shoulders of those who came before them, who are all involved now in pushing the frontiers of medicine through the applications of genomics. And for that, I will continue to remain quite actively involved. So thank you for your attention. I look forward to the discussion. All right.